And welcome to the Invasive Mussel Collaborative webinar series. We have an excellent speaker who has agreed to share his work with us today. Presenter Darrin Hunt with the University of the Ozarks will be sharing his work regarding the duality of shell deposition, how zebra mussel shells introduce a novel substrate in urban and degrade habitat in rural streams. My name is Nicole Angel, and I'm joined by my colleagues Sam Tank and Teresa Gruninger with the Great Lakes Commission. And on behalf of the Invasive Mussel Collaborative, we want to thank you for participating in today's webinar. So before I turn the presentation over to our presenter, I just wanted to provide some background on the collaborative. So the Invasive Mussel Collaborative was convened in 2015 to identify opportunities to advance scientifically sound technology for the management and control of invasive dreissenid mussels in order to produce measurable ecological and economic benefit. The Invasive Mussel Collaborative published a management strategy in 2018 to reduce the invasive mussels and their negative impacts. So to make progress on these goals, the collaborative established 4 different work groups: the research work group, dreissenid toxicity testing work group, coastal site priorities work group, and the most recently established planning and implementation work group. Next, I'm just going to briefly highlight each of these work groups. So the research work group was charged with coordinating existing research and developing targeted plans and projects that address Great Lakes management priorities. So the work group released a mapping tool called the Dreissena Project Coordination Mapper, which you can see on the screen, and it allows researchers and managers to share their work and collaborate with others for the advancement and protection of waterways from invasive mussels. The research work group is currently soliciting additional research and applied control projects, which can be submitted directly through the mapper using that link on your screen to access the mapper. Next, the Dreissenid toxicity testing work group is charged with reviewing the strengths and weaknesses of mussel toxicity control methods used across various labs to identify and make recommendations to further refine and standardize these methods. The studies reviewed for this work have been compiled into a table that provides a searchable collection of literature for Dreissenid laboratory toxicity and open water control testing methods, and is available on the IMC's website. Additionally, a manuscript detailing the recommendations made can be accessed via that bit.ly URL on the screen. Next, the Coastal Site Priorities Work Group was charged with reviewing information on where dreissenids are impacting Great Lakes resources, evaluating sites based on this information, and developing and implementing a system for prioritizing sites for the potential management of activities. The work group released an interactive dashboard and map that displays near-shore data from the Coastal Site Priorities Index. This tool is called the Great Lakes Experimental Mussel Suppression Site Screening Tool, otherwise known as GLEMST, and it features a dashboard and associated indices, which are intended to inform future applied dreissenid research and management activities by aiding site prioritization. And then finally, the Planning and Implementation Work Group is charged with developing a research and management roadmap for the site and regional scale mussel control tools, in addition to identifying stakeholder and funding priorities and remaining knowledge gaps to guide future dreissenid control activities. This work group is building on the products and outcomes of all the other 3 work groups, and you can be on the lookout for more exciting progress from this group over the next year. So today's webinar is part of a communication network facilitated by the collaborative. The purpose of these webinars is to facilitate learning and information sharing on topics of common interest, and the webinar will be recorded and archived along with past webinars and be posted to the Invasive Mussel Collaborative's website. If you'd like to learn more about the collaborative and are interested in staying in touch with our communication networks, we do encourage you to visit our website and join our listservs. And while we strive to minimize tech issues during these webinars, we do apologize for any that might arise. Before we get to our presentation, I just want to cover a few logistical items. So we've automatically muted all attendees to reduce interruptions. Attendees will remain muted with webcams disabled throughout the webinar. However, we do encourage and will accept written questions from attendees during the webinar. So if you have a question, please use the Q&A function rather than the chat function to submit your questions. And questions can be submitted anytime during the webinar, but will be addressed after the presentation has been given. And if you're experiencing any technical difficulties, please use that chat box or email my colleague Sam. Her email is up on the screen. So without further ado, it is my pleasure to introduce our speaker today, Darrin Hunt. Darrin is a freshwater ecologist researching how biotic communities respond to broad-scale anthropogenic stressors like urbanization and climate change. Specifically, he studies how stream benthic macroinvertebrate communities are impacted by habitat changes caused by invasive bivalves in urban and rural environments of temperate and tropical climates. Darrin is interested in bridging the gap between science and the public and has worked extensively with citizen-led bioassessment and management programs throughout the Great Lakes region and central New York. Recently, Darrin has been interested in the green workforce, advising nonprofit organizations on urban flooding mitigation strategies, including green stormwater infrastructure installations. And with that, I will end my slides. And Darrin, you can feel free to pull yours up and we can get this presentation started. Thanks for joining us today, Darrin. We're very excited to hear about your research.
Hey, Nicole, thank you for the introduction. Much appreciated. Let's get this thing going. Good to see everybody. Thank you very much. Great to be here with the Invasive Mussel Collaborative. Great programs, excellent way to enhance science communication amongst the mussel community and interested parties. Really feeling privileged to be here today. As Nicole mentioned, my name is Darrin Hunt. I'm an assistant professor at the University of the Ozarks. This presentation has much to do with work that I did when I was at Wayne State University in Detroit proper. Thus, I am co-authoring this with Dr. Donna Cashin at Wayne State. The name of the presentation is The Duality of Shell Deposition, How Zebra Mussel Shells Introduce a Novel Substrate in Urban and Degrade Habitat in Rural Streams. But before we get started with that, I'm going to kind of talk a little bit about a recent article that was published over the summer that sort of led us to start thinking about how urbanization impacts shell deposition and how native faunal communities respond to that. So let's get cracking. We're going to start a little bit with a bit of background. I know we are in the context of this presentation. We probably have a bit of this background, but just to remind us, like, what invasive species in the Great Lakes is kind of all about and some of the deleterious impacts that we see here. Of course, invasive species can cause ecological, environmental, economic damage. They've been around a long time, sort of came in, the broader thinking is they came in through the Welland Canal in the 1800s, which is later expanded into the St. Lawrence Seaway. It's kind of why you see this at your bottom right, this picture here, sort of showing the breadth of invasive species and the broadness of the lake and lake system. So yeah, it's a lot of pathways for organisms to enter here. Since the inception of the St. Lawrence Seaway, there's been more than 180 non-native species introductions. Thankfully, not all of them have established, but many of them have. Of course, we're here to talk about invasive bivalves. They're one of the most damaging and expensive, costly to manage organisms that are being dealt with in the Great Lakes right now and have been for, close to 50 years at this point. So 'tis the reason for the season. We're here to talk about invasive dreissenids in the Great Lakes. Of course, zebra and quagga mussels. You see zebra mussels on your left there. There's a picture of the organism Dreissena polymorpha and its broad coverage throughout the Great Lakes region. Next to it, you see the quagga mussel, Dreissena bugensis, also expanding greatly throughout the lakes. I updated this presentation recently, and in the previous time that I had given it, there was few quaggas in Lake Superior, and now it's completely covered, right? So just within 5-6 years, we've seen this massive increase throughout the lakes. They're thought to have moved in through the ballast water of ships in the 80s. And interestingly enough, you're probably looking at these two range maps and thinking, one of these is just everywhere and one of these is just somewhere. I think we're learning more and more about these organisms and how they spread and the habitat they like, but a lot of them could be selected by habitat, right? So there's some preference differences between the two species. Zebra mussels tend to favor shallow waters, can cling to hard surfaces, but quagga mussels can get pretty scary because they don't really require that hard surface. They can exist in cooler temperatures. But And we have seen them in river systems, came across them over the course of this research, certainly. But though this is an empirical observation, it seemed that in these sort of tributaries, the sort of faster moving tributaries of the Great Lakes that I kind of wandered into, I noticed a lot more zebra mussels than quagga mussels. It'd be interesting to see how that changes over the next several years. I know that we kind of understand that dreissenids move into rivers, especially big rivers that kind of behave a little bit more like lakes. But I think largely they're sort of thought of as a lake organism. If you're looking at this little GIF map that I have off to the side, this shows the invasion pathway throughout the United States from 1986 to 2011. Clearly, it has expanded beyond that within the past 10 to 15 years. But largely, we think of them as sort of a lentic organism, right? They like sort of slower moving stuff. And it has a lot to do with the reproductive pathways. But they've been found in large rivers throughout North America. Establishment is a little bit less likely in shallow, fast moving streams, but we have seen them there. It does happen. When they do invade, they are susceptible to population crashes. And as the animal itself sort of deteriorates, and which happens relatively quickly, the shells that they inhabit are left behind. So these shells can be deposited in the environment behind them as these populations flux and move throughout time, and can increase the interstitial space and surface area of stream bottoms, which, of course, we've seen this in lakes, but there's very few studies addressing how smaller wadable streams, a little less than 30 meters wide, would respond to this change in habitat. So when we think about the consequences of bivalve invasions, the first thing that always comes to my mind is habitat degradation. And if you're looking at this picture that you're seeing here to your right, with the two organisms that we're sort of talking about today to your left, Bugensis and polymorpha, that's actually a photo that I took of some substrates in the Rouge River. So as you can see, the substrates are mussel shell deposition. Like that is what we're seeing there. So it's kind of interesting to think, these organisms or these systems that are so dependent on habitat diversity, what happens when that habitat becomes completely obscured with mussel deposition? And how do those organisms that are selected for by that interesting and diverse habitat and stream systems respond to this homogenization of habitat? So as we know, living and dead Dreissena species can biofoul lake and river substrates. Insofar, they increase habitat complexity and can, of course, cause habitat losses. But that's not all. Clearly, we can see loss of invasive species. We have a unionid mussel in this photo here, maybe a fat mucket or something like that. They're in competition for other filter feeding bivalves in the system. They can even colonize those organisms. And of course, this leads to reductions in biodiversity overall. There are also massive economic impacts. We've all seen, heard, and read stories about various bivalve populations and communities occluding effluent pipes of power plants and so on and so forth. This costs a lot of money to manage. There's a lot of numbers out there. I tried to find one that was a little bit more conservative, but it's estimated that about $500 million are being spent per year to manage these organisms in the Great Lakes region. So that's a lot of money that's going into doing this. So resolution is the challenge, of course, but obviously organizations like the Invasive Mussel Collaborative are working on just that. So kudos to you all. So when we're talking about how stream organisms are impacted by this phenomenon, the stream community that we're sort of interested in is the benthic community. The benthic community is the community that kind of hangs out in stream bottoms. So we're talking about macroinvertebrate organisms. We're talking about fish. They're ecologically important. They have functional roles where they transform nutrients and send them downstream. So there's this complex environment that's being dealt with in stream ecosystems. In these photos here, you can see, of course, some flowing waters, a bubbling brook that we're all happy to see. And then you have some sunfish here, some bass, of course, and then caddisflies, right? So benthic communities are really susceptible to these changes, right, that might happen. So pollution, sedimentation, stream channelization, habitat degradation caused by invasive bivalves, and so on. We can estimate that there's going to be something going on here. So we got to kind of figure out what it is. This is especially because macroinvertebrates and fish are used as a proxy for water and stream quality. So citizen-led bioassessment organizations tend to lean into this faunal group to try to figure out what's going on with the water quality. And there can be a lot of confounding factors, as we can sort of see here, right? We're talking about habitat changes, we're talking about urbanization, all kinds of issues that sort of work together to change how this community might respond. So we're going to chat a little bit about that. We have two research questions that we're going to be discussing today. One is, of course, how do invasive dreissenid shells alter stream benthic habitat and impact macroinvertebrate and fish communities? That's question number one. Question number two is we're going to kind of take a deeper dive into urban and rural systems and how that might impact them. So the second one, of course, is do macroinvertebrate responses to invasive mussel shells differ in urban and rural streams? So first, of course, how do invasive mussels or invasive dreissenid shells alter stream benthic habitat and impact macroinvertebrate and fish communities? Okay, so we looked at two major rivers in southeastern Michigan, the Rouge and the Huron rivers. These are urban rivers with expansive streams, creeks, tributaries, cool headwaters, and sort of expand as they move downstream, and of course, run into the river eventually. They service the Detroit metropolitan area. Not only do they have a strong fishery economy, but they're used for recreation, hydropower, etc. So this is a close look at the two watersheds in question. To your left, you see in green the Huron, and to your right, you see in red the Rouge. There were 16 total sites selected here, so 8 invaded and 8 reference sites. And if you're curious, there are also 8 sites per river watershed. Within each river, there were 4 invaded sites and 4 reference sites. What I mean by that is there are 4 sites that we found invasive mussel shell deposition in, and we wanted to contrast that with 4 additional sites. So if you're looking at the map, you see yellow triangles. Those yellow triangles are the invaded sites, and the black triangles are the reference sites. We did a one-to-one match on each of these using habitat assessments. So the metric we used to assess habitat was the qualitative habitat evaluation index, and we made sure that the scores when comparing these particular sites, references and invaded sites in a one-to-one way, that they were within 10% of one another in terms of their score. Now, if you don't know about what the qualitative habitat evaluation index is, it takes sort of a broad look at many different factors, both in stream and out, to determine habitat quality within a reach of stream. So we did this for each one of these streams and just made sure that there wasn't too many other confounding variables, trying to find ways that we can isolate the impact in a natural survey of dreissenid shell deposition. I also think it's worth noting that these river watersheds are sort of interspersed with reservoirs and lakes. So in most cases, there was some form of upstream source population, potentially, either mussels that were existing at some point within the lakes that were directly upstream from the stream sites, or also in some cases, in one case specifically, we found living mussels in our site as well. But the objective here was not to look at how living mussels impact stream benthic communities, but rather looking at how the complexity that's added by mussel shells might contribute to some form of selection for particular community composition and assemblage. Okay, so how did we do it? The first thing we had to do was quantify shell coverage, right? So we used a quadrat system for that, which was a 0.5 meter squared PVC frame, which we tossed in triplicate. So we did, 3 replicates in each site in affected areas. And as you can see, by looking at the sort of pictures below the quadrat and enamel pan, which you're seeing to your upper left, you can see like that there is a lot of shells, right? We removed all the shells within the quadrat area, rinsed them in a sieve, and cleaned them up and counted them. So we had a lot of students working really, really hard to try to quantify exactly how many shells we were seeing in each one of these sites. That was done in the spring. Later in the summer, we came back, we collected and identified benthic macroinvertebrate organisms. So this would be like the entire breadth of the benthic macroinvertebrate community in each of these sites. We did that once in the summer and twice in the spring and again in the late summer, early fall, we collected and identified fish. We did this using a seine net, seine for about an hour, kept our fish in a bucket, and then, of course, identified the fish to species. But yeah, some pretty cool pictures there. If you're curious, there is a red-side dace in the lower right-hand corner of your screen, which is a critically endangered fish. I always try to include that picture anytime I can in these presentations because, hey, red-side dace are cool. And critically endangered fish, we should know a little bit more about them. So yeah. All right, so the first thing that we wanted to sort of nail down was how do mussel shells contribute? Is there a statistical difference? And okay, I get it. This is sort of a priori information. Of course, if there's invaded mussels in the system on top of our sort of native mussel shell deposition, we're going to have more. And that's exactly what we find. No surprise here. In all of our sites, regardless of whether or not they were invaded or uninvaded, we had our native faunal deposition, right? So we had gastropod shells. There were, of course, unionid mussel shells there. But once you added the dreissenid shells on top of it, it just expanded exponentially, right? So the shell deposition that we would see in those sites was really, really high. And of course, there was a statistical difference. We did like just sort of a general comparison using a Mann-Whitney test. And of course, it was super significant. But something that we did want to see is like, okay, if we're having this happen when we just are comparing our invaded and uninvaded sites to one another, how do they differ between each river system, right? So we compiled all of our Rouge sites together, uninvaded and invaded, and our Huron sites together, uninvaded and invaded. And we compared those, and we saw that there was not a statistical difference between the way that these shells were being deposited within each stream. So this allowed us to sort of feel confident as we move forward with our assessment, knowing that at least as it relates to shell deposition, we're not seeing differences in habitat within the river, because that obviously would impact our stream benthic community composition if what we're saying is they're selected for by habitat. Okay, so taking a look at how macroinvertebrates responded to increasing shell densities, I should note, and if you've checked out the paper at all, that we went through and compared the invaded and uninvaded sites using nested ANOVAs. A nested ANOVA is sort of an ANOVA that just assumes there's no interaction between the two groups, and the two groups are the Huron River and the Rouge River, right? So in general, we wouldn't really necessarily expect any interaction between those two systems. But alas, as it relates to the multitude of metrics that we ran, various diversity measures, looking at evenness throughout the community, we did not see a bunch of significant difference, right? So this is just kind of looking at invaded versus uninvaded. But we noticed as we're looking through all this stuff that the shell density was not completely consistent amongst these sites. And it actually increased along sort of a gradient, right, between invaded and uninvaded sites. The uninvaded sites had a mixture of shell densities ranging from 0 shells per meter squared up until to almost 100. And then, of course, our invaded sites had many more shells than that, but also existed along a gradient. So not every site we went to was entirely dense with shells. So we decided, it's probably a good idea to see how these organisms are actually responding to a gradient of shells, right? So like along varied shell densities. So we used an ANCOVA to do that. We grouped up our rivers, of course, by Rouge and Huron, and then used the actual shell density, which was a gradient from 0 to over 7,000 in many cases per meter squared, to assess how these organisms are responding to shell density along this gradient. So it's kind of yielded some interesting results. We'll go through some of those now. One of the things that we tested was the Hilsenhoff biotic index. If you don't know what that is, it actually is a metric that kind of takes macroinvertebrate communities and makes an estimation on organic pollution and so on. So there was a significant difference between river and that's great and good to know, but it's not really relevant to our study. We really want to see how shell densities, as they change along this gradient, are impacting our organisms. But we did see a difference when we're looking at sensitive Ephemeroptera, which are mayflies, Plecoptera, which are stoneflies, and Trichoptera, which are caddisflies. So we did see a significant difference there along this shell density gradient. Similarly, in our benthic index of biotic integrity, which is similar to the percent EPT test, which is looking again at mayflies, stoneflies, and caddisflies. This test looks at each one of those population or orders individually, and then also looks at things like overall family richness, looks at things like species dominance too. So we had some significant results in that as well. So taking a look at everything together, when we pooled between all rivers, we saw that the EPT relative abundance decreased as shell density increased in the Rouge River. Now, this is in conflict with what we know about lakes, right? Lake systems generally with increased benthic habitat complexity tend to increase in abundance and potentially diversity across an increasing shell gradient. We saw the exact opposite in wadable streams. And it's kind of an interesting thing to think about. But we were curious, does this happen in the Rouge and the Huron the same way? Are each of them significant? So we've looked and we found that our relationship along the trend line, meaning this is the R squared really, and how strong the relationship is, this degradation of these three orders as shell density increased, was much stronger in the Rouge than it was in the Huron. This is the Rouge River here. This is the 8 sites that we're seeing. And we did not see a significant relationship in the Huron, but we did see a significant relationship in the Rouge. So as shell density increased, we saw fewer and fewer mayflies, stoneflies, and caddisflies. And these are some of the most sensitive gilled organisms that we see in the benthic community, and sort of central to what bioassessment is based around. We kind of look at how those organisms respond to various stressors. Conversely, when we're looking at the BIBI scores, again, reminding you that this also looked at Ephemeroptera, mayflies, Plecoptera, stoneflies, and Trichoptera, caddisflies. And we did see a degradation here as well as in BIBI scores as shell density increased, and did the same thing. We wanted to isolate it and see kind of what's going on specifically here. In contrast to what we found in the Rouge, it was actually the Huron that drove this here. So what could be doing that? In thinking about that, we sort of got to this point where it's like perhaps a broader community of benthic macroinvertebrates outside of our sensitive gilled organisms could be driving this as well, even though we didn't see significant results as it relates to diversity, right? But something else is driving it. But interestingly enough, when we're taking our sort of zoom out message on all of this, we're sort of seeing that regardless, these wadable streams that have this complex benthic habitat are responding really, the communities that are occupying those habitat are responding differently than what we're seeing in lakes. So that was a very interesting thing to sort of consider. So everybody's wondering about fish, right? Well, we didn't really have anything significant to say about fish, so I say fish slipped away. Fish communities did not differ by river, and we added a factor where we compared season and dreissenid shell presence. And I really like this picture. This is one of our technicians that we worked with extensively on fish surveys, and if you look at this, the 2 northern hogsuckers that he's holding, one is literally slipping out of his hands into the net. So it's kind of interesting to see, of course. Okay, so what are our conclusions on this first study? And this is, again, the one that we saw, or that was recently published in July. Sites invaded by dreissenid mussels had higher shell densities than uninvaded sites. Habitat losses following mussel invasions led to fewer sensitive macroinvertebrate taxa, but did not impact overall community composition as shell densities increased in both rivers. And mussel shell deposition didn't have an effect on fish communities across rivers or seasons. So in general, let me see if I can get this little weird window out of my way, that the broader significance is the legacy of invasive species can persist after populations crash, which they frequently do, or while in flux. So that's looking at some of the first work that we kind of engaged in these systems. And it led to a lot of other questions. One of the main questions was, okay, we know that we have an urban system in the southeastern Michigan. There's a lot of consequences to that. Early consequences of land development are deforestation, impervious surfaces, pavement structures. Of course, there are resolutions for that, but they're only now starting to be incorporated, pervious pavement and so on. Stormwater runoff and wastewater discharged into streams. So this is just kind of some of the normal stuff that we deal with when we're thinking about how urbanization impacts stream benthic communities. Subsequent problems, of course, flooding, sedimentation, stream channelization, water table changes, habitat alteration, and of course, degradation. These stressors that are associated with urbanization are known to impact benthic communities and invasive species, right? So this is just causes a whole suite of issues. And of course, being someone who studies how geomorphological changes and either whether they're biotic or abiotic are impacting stream funnel communities, this is of great interest to me. So how do we get to this? So this is now moving into our second question. Do macroinvertebrate responses to invasive mussel shells differ in urban and rural streams? Okay, so the geographic scale is similar to the first, right? We have our Huron watershed all the way to the left in green, our Rouge watershed in red, and then of course the Clinton we have added here to sort of round things out and add an additional urban watershed to the system in blue. And of course, this is all in southeastern Michigan. Here are our study sites specifically, and you're looking at the ESRI world population density estimate layer. If you're looking at the map itself, the triangles are our rural sites and the squares are our urban sites. If you're curious, what was our metric? We kind of looked to see how much was light urban and how much was rural or forested in these particular sites. To qualify these sites for this study, the urbanization level, meaning it had to be light urban, over 85% of the 1 kilometer polygon that we drew around each site. And likewise, it would have to be 85% or greater rural or forested in order to qualify as a rural site. There were 6 total sites. 2 were in each river watershed. 1 was rural and 1 was urban. In order to kind of get to this, we used data that's publicly available, macroinvertebrate biomonitoring data that was offered up by the Clinton River Watershed Council, the Huron River Watershed Council, and of course, Friends of the Rouge, who we worked with really closely on several projects. Another thing, how do we get to this urban question was we decided we were going to actually introduce shells into the system. And in order to see how the existing macroinvertebrate communities would respond to that, there had to be no record of dreissenid invasion in these reaches of river. So we inspected to make sure that there were no shells there. And of course, as I mentioned, the sites were sort of qualified using existing data, GIS layers from S3. Okay, in order to do this, how to introduce shells in a responsible way to a system, right? Obviously, we can't just go and pour a bunch of dreissenid shells. It would be a hard way to control it. So we created macroinvertebrate mesocosms, built these in my garage using a PVC frame, 4 plastic containers filled with whatever colonization substrate we decided and covered in plastic mesh. So the colonization substrates were 0% shells and 100% cobble, right? So cobble trying to mimic like what is a normal substrate in a stream system, what you would expect to see there. We also had a mixtures between shells and cobble. So 75% shells and 25% cobble, 25% shells and 75% cobble, and then we had one chamber that was 100% shells. These were assembled with this plastic mesh so that organisms could come and go, but there were also 1-inch wide holes that were bored in the sides on all 4 sides of these chambers so that macroinvertebrates could actually move through the chambers and select whatever habitat that they seemed to favor. these actually would end up being buried in the stream system, right? So you can kind of get a sense of how they look. And for your guys' information, just so you know, they weren't arranged this way necessarily within each rack. The arrangement of treatments or chambers or trays within each PVC frame was entirely randomized. So we sort of drew straws for that. Okay, so this is some pictures of the macroinvertebrate mesocosms after they were implanted. Up at the top, you see some of the mesocosms just sitting there. We did 10 replicate racks per site, right? So we made a lot of these. Anchored in place for a 40-day colonization period. And then, of course, after 40 days, we went out and removed the trays from the river. Stream side, we separated the chambers from the trays and kept them separate from one another, preserved the contents until we would later take them back to the lab and do a family level assessment. If you're looking at the picture in the center of your screen, or in the center right, rather, that is a handful of these mesocosms in place. You can see one is here, one is over here, and one is sort of upstream here, or downstream. Yeah, upstream here. So yeah, we kind of tried to intersperse 10 of these throughout each reach or river. And at the very bottom, of course, this is my partner in crime, and who helped me pretty much remove all of these things from all of these sites, which was a pretty arduous journey we were on for that. But again, always a good time, science in the field. Okay, so what are the results? The question, of course, do macroinvertebrate diversity differ by river or urbanization or among shell treatments? We looked at Shannon diversity, and we used a split plot ANOVA to do this, which kind of acts like a randomized block test. In general, it looked at 4 factors: river, urbanization, shell treatment, and effects of interaction. And again, we're just looking at diversity here. No surprise, there was a difference in diversity between rivers. We kind of knew that going in. We saw this in our last analysis. Urbanization for diversity, there was no impact of urbanization. And basically what this means is, looking broadly, our urban group didn't differ from our rural group, or our rural group statistically. Nor did we have that issue amongst shell treatments as it relates to diversity. But something that I found very, very interesting is that we did have an effective interaction. And okay, looking at split plot ANOVAs and trying to assess what exactly an effective interaction is going to tell us it can be a little bit tricky. But I always sort of think of it as some response variable is behaving differently in light of another response variable, right? So shell treatment or urbanization is responding differently between the groups, or it really just kind of depends on how things are grouped. So looking at this interaction p-value, we kind of looked at a few different things, right? And one of the things that we noticed was the difference in diversity among shell treatments was dependent on urbanization, right? So whether or not you had a urban system or a rural system dictated how the macroinvertebrate community was going to respond. And this can be pretty interesting, right? And if you're looking at the graph right now, if you see the sort of gray line that has the circle, that's sort of moving upward, that is our urban system. And we had a significant difference when we took a look at the pairwise comparisons along the linear mix model, that there was a difference between the 25% treatment and the 100% treatment. So our more high density shell treatment was colonized more by the stream benthic community than the 25%. But when you look along the sort of entire line plot, you can see sort of a general favoring of upward movement. Although there was no statistically significant difference in rural chambers, it did sort of degrade a little bit. And so I kind of think about this as the bugs were sort of happy to see that there was dreissenid shells in our urban system, but they were a little bit sad to see them in the rural system. So why? Why would this happen? It's sort of an interesting thing to think about. Knowing what we know about urban systems and degradation of urban systems, we know that many of the stressors, like sedimentation, that are indirect effects of other things like stream channelization and so on and so forth, urban development, etc., that we expect there to be some habitat loss. So introducing these shells to the system sort of could have potentially introduced a new benthic complex habitat that these organisms favored in terms of colonization. So it's kind of interesting, right? Because we didn't see that in the rural system, where we have lots and lots of diverse habitat. Okay, So before we move on beyond diversity into the relative abundance of these different organisms, I wanted to point something out. As it relates to diversity, we have a particular system that sort of outranked the rest, and that was the Huron. This is not specifically relevant as it relates to diversity, nor is it really what we're asking in terms of these questions. We're looking at urbanization, and we're looking at how these organisms use shells in the context of urbanization. But something that's going to come in just a minute and sort of may be a driver for the differences that we're going to discuss in a moment is that the Huron was the most diverse of our systems, right? So we're going to move on to looking at macroinvertebrate relative abundance. We're asking the question in this context, do macroinvertebrate communities and shell treatments vary within rural and urban sites of the river? So when I'm talking about within, what we're no longer going to be comparing anything amongst rivers. We're going to be looking at what happens within each site, as far as like how the organisms choose to use the colonization substrate. Of course, the Huron, if we're comparing relative abundance of the Huron to the Rouge to the Clinton, there's going to be differences. We know that. What we want to sort of dissect is get into what happens within each mesocosm chamber or rack that has the 4 treatments. So the Rouge urban site had no specific difference, very urban space. The Rouge rural site had no specific difference, again, very urban space. Clinton urban, no specific difference. Same thing with the rural. Now, when we get to the Huron, things are a little bit different. Of course, there's no difference in the urban, right? That we have our degraded communities, probably not a bunch of diversity hanging out in there. So that's not significantly different either. But we did see differences when we were looking at the very much most diverse reach of river in our most diverse river system, the Huron. So this is kind of an interesting thing. We did a Permanova to do this, and I'm happy to chat with anyone after the talk about stats. I like to chat about that stuff. So sharing is caring. So if you want code or to talk that stuff over, we can connect. In doing a Permanova, one of the things that you want to do is you want to see what are the major contributors to these changes. There's a number of ways to do it. You can use ANOSIM, which is a modeling program. You can also use SIMPER, which is looking at similarity percentages. And that's what we did here. But we were trying to figure out like macroinvertebrate relative abundance differed between the controls, which is 0%, and the high density shell treatments, right? So along this gradient of 0 to 25% shells to 75% shells to 100% shells, we saw a strengthening of interaction, right? So there was no significant difference when we were comparing the 0 to 25% shell treatment. But when we talked about the 70 or looked at the 75% and the 100% shell treatments, we did see significant differences. And the p-value decreased, sort of adjacently insinuating that there was a strength of response. So the similarity percentage metric kind of looks at how similar these populations are to a control, right? So we're comparing the control to the 75% and to the 100% in each column of this table. And there were really 6 families that contributed to this, and these are macroinvertebrate organisms, right? So Chironomidae, which are essentially your non-biting midge flies. These are really, really abundant in all stream systems. So it's no surprise to see that they're there. But there might be some changes they're driving some changes in these systems, which is kind of interesting. Then there is Leptophlebeidae, which is a mayfly, Hydrocychidae, which is a caddisfly, Heptageneidae, which is a mayfly, Almidae, which is a river beetle, a diving beetle, and then Ephemeroptera, which is another mayfly. So you see a lot of sensitive organisms in this list, right? A couple of tolerant ones, but mostly driven by some really, really sensitive organisms. So let's take a look at each one of these, looking at how chironomids differed among shell treatments in these rural reaches of the Huron. And the 0 and 25% treatments, meaning the ones that had no shells or only 25% shells and 75% cobble, were less colonized by than the 75 and 100% treatments, right? So keeping in mind that this is all in one reach of river. So in these chambers, the chironomids, which are, of course, again, non-biting midge flies, were tending to favor higher density shell treatments. And this kind of makes sense if we know a little bit about what chironomids are up to when they're nesting. They try to find these sort of like interstitial complex spaces to hide from prey. So no surprise that we would see more of them when there's more of those spaces to occupy. And that is interesting. But one of the things that I found really interesting is how our sensitive organisms are responding to these shell treatments. I'm first, when we're looking at this is our Ephemeroptera, as you can kind of see here. This is our mayfly, and then our Trichoptera, of course, which is our caddisfly. In this list of four different types of organisms, Hydrocychidae is the only caddisfly, right? So this is the one that's in green. And if you know anything about caddisflies, especially hydrocychids, they're net spinning caddisflies, although the limnophyllid in this picture would insinuate otherwise. But they're net spinning, they don't build cases. So they need to have a very stable benthic environment to do so. So one of the things that I was thinking as a possible driver for this is that even though shells introduce like a more complex new environment for organisms to colonize, they're a little unstable and they might move with the hydrology of the water. And if that is so, they could potentially break up the nesting sites and collecting sites for downstream stuff that caddisflies are usually lying in wait for. So you can see like a degradation of this family over time, where it's about 12%, about 13% of the population in the control, and then it sort of degrades until it reaches just about 5% of the population. So this again is sort of like the amount of these organisms that we see in this environment, percentage-wise, right? Like how representative of the community are they? So now looking at the other three, which are mayflies, we see conflicting responses. We see this leptophlebid in blue that just continues to rise throughout shell treatments. They're more represented with more shells, which is sort of interesting because they're so sensitive. But we'll get to talking about that in a minute. I want to focus on the other two, Heptageneidae and Ephemerelidae. Like caddisflies, these are organisms that love to inhabit that stream substrate, right? They're Heptageneidae are scraping organisms. They are trying to eat that biofilm and periphyton off that is grown on the stuff that's hanging out in the substrates of the stream. So clearly, this is doing something that's sort of challenging them a little bit. If we have an unstable stream bottom, how much periphyton can grow, how much biofilm will accumulate on these substrates, and does it make a good feeding ground for heptageneids and ephemerelids, which are mayflies that interact with that bottom. Interestingly enough, there's a lot of reasons we could think about what's happening with the leptophlebiids. They swim around a lot. They're fast-moving little organisms, and now we're seeing that some of the organisms that are occupying this space are vacating the premises as the shell densities increase. That could, one, not impact leptophlebiids as much because maybe they're a little bit more motile and like to move around a little bit more, or two, that we see a change in niche overlap. The ecological shifts in resource distributions could change too. If what we're talking about is what's happening in biofilms and periphyton as it relates to shells and shell densities, this could actually have an impact on the overall Ephemeroptera, Plecoptera, and Trichoptera community, which are our sensitive gilled benthic macroinvertebrates. Okay, so kind of winding up into our conclusions, our urban system has low habitat heterogeneity, right? Through the stressors of urbanization, it's lost habitat over time. In our rural systems, we have high habitat heterogeneity. Low habitat heterogeneity has led to and selected for low diversity and abundance of macroinvertebrate organisms. Conversely, high diversity and abundance is seen in our rural systems that have a lot more habitat to occupy. Enter Dreissena shell deposition. At the lower levels, we don't see much difference, right? No one really seems to care too much in either system type. But as you start to add more, you start to see increasing diversity and abundance because it's potentially adding habitat to our urban systems. conversely, we see potentially the exact opposite. We see this slow degradation of macroinvertebrate communities as habitat becomes obscured in these rural systems. And then eventually, once those densities become so high, we start to see this general shift between dominance within these ecological systems. So in words, ecological communities. In rural systems, high densities of dreissenid shells inhibit colonization by sensitive gilled systems or gilled organisms. In urban systems, macroinvertebrates favor substrates with high densities of dreissenid shells. So we see an opposite response, which is that's news, right? This is a novel finding. Rural and urban macroinvertebrates communicate, or communities respond differently to dreissenid shells in rivers. With that, couple of familiar names on here that I saw in the attending the webinar today. Thank you to everybody who helped me get this done. Thanks to my funding organizations, and of course, this project was sort of funded adjacently by the NSF. So, if anyone has any questions, I would be happy to take them.
Thank you so much. That was really amazing. I really appreciated all your pictures. They were great. I especially love the little fishes you put in there. Red-side dace and hogsucker are two of my favorites.
Classics.
So that does conclude our presentation for today, but we're going to move on to answering some questions. I see that there are a few questions rolling in. We have one question. Was there a difference? This was referring to the beginning portion of your talk. Was there differences in terms of round goby between systems?
Interestingly enough, we literally found a single round goby when we were sampling for fish. So we didn't really have a very good representation, which is interesting because in this area of the Rouge, there were a lot. In fact, one of my colleagues was doing a study on that. I understand the nature of the question for sure. Is there some kind of coupling between two invasive organisms from the same region? Unfortunately, we didn't have representation of round goby that would allow us to make claims about that in our samples.
Interesting, yeah. I feel like we see round goby a lot in lakes and lentic systems, so it's interesting that they're maybe not as abundant in the rivers and streams. Or
to kind of kick back on that a little bit, we know they're there. Lab mates were actually finding them. We had some shared sites, and we were actually seeing them there too. Just in my particular, I don't know if it's the times we went out in pretty early spring. We were trying to get spawning periods, right? So that was kind of the idea, is we wanted to try to get as much spawning representation as we can, right? Because in general, we're asking questions about how fish are using these substrates for the normal stuff fish use substrates for, right? So we wanted to see if we would see, sunfish that were creating that, breeding nests in different periods of the year. So it might have been the time that we were out. I'm not exactly sure, but we only saw a small handful.
Interesting. Yeah, I know round gobies are also good at evading certain fisheries techniques, so maybe it was that as well.
Yeah, it could be, right? Making too much noise in the stream.
Yeah, looks like we're still waiting for other questions to roll in. I did have one question.
Sure.
Where were you obtaining the mussels for your little mesocosms?
Like specifically?
Generally, whatever you'd like to share.
Yeah. So the impetus for this suite of projects as a whole is what we saw at the outflow of Newburgh Lake in the Rouge River. There had been the lab that I did my PhD in had used this site as a collection site for zebra mussels, right? So we did a lot of in-lab tests looking at oxidative stress and other things like that. One of my colleagues was collecting their organisms from this site. And when I went there to sort of do some similar stuff and noticed that it's pretty much just shells. Everything had died. And over time, it never really replenished. And this is the site where I actually found like a single living zebra mussel there. That was the only living organism we found at any of our sites. So what we ended up doing was we collected shells for mesocosm enclosures from that site. And the process was is we'd pull them out by the 5-gallon bucket. We just could pretty much shovel them from the stream bottom into our buckets. We took them back to the lab, soaked them in 90% ETOH for 24 hours, rinsed them, soaked them again, dried them, and then brought them into our mesocosms and then again into our experimental sites.
Interesting. I would never imagine like just that many quantities in a moving system. But
if you haven't checked it out, like it is definitely worth the trip just to put on some waders and walk around in the outflow. You can feel them crunching underneath your feet. I tried to dig to where they ended, and it was essentially I gave up after getting about a foot deep. I was like, you know what, how deep am I to dig in this stream?
looks like we do have Andrew with his hand raised. I did enable your mic and camera, Andrew, if you'd like to go ahead.
Yeah, awesome talk, Darrin. Really cool stuff. I was just wondering how do you think your results would have differed if you would have used like live juices of zebra mussels? Like, do you think the deposition of pseudofeces would have significantly changed the results of this study?
That's of course a really tricky question, right? Thanks, Andrew. Good to see you, by the way. Yeah, I of course pseudofeces is going to put a plume of available nutrients into the system, create opportunities for biofilms and periphyton to increase, which could deeply shift the community composition of the benthic macroinvertebrate organisms that are present. And depending on how much you're seeing in terms of overlapping resource usage and subsequent partitioning amongst communities, I would think that it would gravely impact the results of this study. I also think that something that's pretty interesting is that if we're working in outflows where presumed living populations or communities of dreissenids exist, that is going to impact the nutrient deposition downstream, right? That's going to be the thing that informs biogeochem of the reach. That's why I think that it's really good to try to get those sites in there that don't have dreissenids for the mesocosm enclosure study, right? Because we're sort of alienating that specific confounding factor of the physiological roles that dreissenids are taking in upstream aquatic systems, right? Could be reservoirs, lakes, or even impacted stream reaches that have living populations in them.
Great. And I think we have one more question to close out. Did you look at any water chemistry differences between streams? A colleague of ours has found higher bacteria levels and lower DO in shell hash.
So yeah, great question. Yes, of course we did baseline physical chemical parameters where we looked at pretty much water quality values. We did not do any nutrient analysis or metals or dissolved organic carbon or anything like that, but we did look at dissolved oxygen. I did run them against one another, and maybe because of the proximity, there's of course a loss of dissolved oxygen that we would anticipate being in urban systems too, but it wasn't a statistically significant difference between our urban and rural sites.
Great. I'm not seeing any more questions rolling in here. We're getting close to our ending time, so last call for questions. Anybody have any burning questions for Darrin? If not, I will just want to say, thank you, Darrin. This was an amazing presentation. And thank you all for joining today. As a reminder, this webinar will be recorded and posted on the Invasive Mussel Collaborative's webpage. And if you have any additional questions or follow-ups, please feel free to contact myself or Sam at the Great Lakes Commission, and we can get you in contact with Darrin or help answer those questions in any ways possible. So thank you all for attending, and thank you, Darrin, for a wonderful presentation.
Great to see you guys. If you have any questions, feel free to reach out. I'm happy to talk more about all this stuff.